Hello. Hi. Welcome to session 3 of day 2. Our first speaker is Filipe Menezes, who is from Helmholtz Center, Munich, and his work focuses on quantum methods for ligand analysis. He's going to talk to us about a novel tool for ligand analysis using quantum chemistry.
So I hope the microphone is working. So hi, everyone. Good afternoon. Extending Gustavo's words, I hope that after lunch, everyone is more amenable to agree with what I'm going to say. As said, I'm Filipe. I'm a quantum chemist by formation, and now I work at the Institute of Structural Biology in Helmholtz, Munich. And today, a few words about one of the methods that we have been developing with CCT for people for ligand validation. So a brief outline. First and foremost, motivation. Why is chemistry still important and perhaps even more important than ever? Then a case study where we're going to show our new tool in pocket analysis. We're going to analyze a specific protein-ligand complex and show you what this tool can give you. And last but not least, we're going to study, or I'm going to show you a few additional downstream applications where you can see all the potential applications that the tool can give it to you. So we start with motivation. And to motivate this whole job or this whole thing, I chose a simple task that is nowadays almost routine, which is to basically use co-folding models, in our case, AlphaFold 3, to predict the structure of a protein. And the protein that we chose is basically cytoglobin B. Now, here we have an overlay of 8 different crystal structures of cytoglobin B. You see that they all overlay really well, not only the secondary structure, but also at the atomic detail, because you can identify that cytoglobin B is a heme-containing protein. You can identify the heme group down there. And you have the iron, the porphyrin ring. And in this case, we have 2 descriptors, which are going to be useful for us. We have 2 histidine residues binding to the heme group up and down. In all the cases that we found on the PDB, all of the histidines coordinate with a nitrogen atom. So we move now to the AI structural predictions. AlphaFold 3 knows somehow cytoglobin exists. The structures are old enough. So we cannot really just ask AlphaFold 3 predict the structure of a known protein. It's going to fetch information from the training, and it's going to reproduce what it already saw. So we introduced a few alterations, and these alterations are just small peptides, so that AlphaFold needs to think a bit what's going to happen when I try to predict the structure of this protein. We did this for 5 peptides, different ones. We predict 25 structures, and this is the overlay of 5 of those structures that we predicted. Now, you identify, or you can appreciate, the overlay of the secondary structures is fantastic. The overlay at the atomic level is also really, really good. You can ignore the peptide. It's not really important for what we want to do. The histidines are all on top of each other. But if you take a closer look, you'll see a few things. First, the lower histidine, and here lower is just our classification. AlphaFold does not know what up or down is. The lower histidine is always n-coordinated in all the 25 models that we predicted. You go to the upper histidine, and things are not so nice, because 6 of the 25 AlphaFold models that we generated, they predict an n-coordinated histidine, meaning 19 of these models are C-coordinated histidines. And I don't know how much chemistry you know, but the C-coordinated histidine is something that chemists struggle really a lot to get in the lab, let alone under biological conditions. So this is complete biological nonsense. Now we can ask the question. One histidine is clearly correct in all of the models. The other one is clearly incorrect in almost all of the models. What is the problem? Where is the underlying reason for this behavior? This is the model. I think we agree, all of us, that AlphaFold is good enough to predict the structure of proteins, and therefore it's hardly likely that AlphaFold is going to screw up so badly the chemistry just because the model is not good enough. Is it the data? Well, can be. We can probe these, and we're going to analyze later a few examples to see if we can find issues in the database, the PDB. Or is it the chemistry? Chemistry is so hard, and an AI model cannot learn the chemistry, and chemists are still safe from this AI revolution. In any case, we all should agree that these methods, they excel at predicting geometric features. They have problems at the chemical detail. So what could have gone wrong? I mentioned the data. Let's look at a few examples. And if you think about globins, it should come to your head, or at least to my head as a chemist, hemoglobin. Hemoglobin contains also the heme group. You can identify this here. We have a tetramer, so we have 4 heme groups. And these heme groups differ from the previous one. The previous one had 2 histidines around the iron. Here we have 1. So we have a mono-coordinated heme group. This also hints that the chemistry is different because in this case, we have iron 2. And this is a high enough resolution structure. And if they didn't screw up with the restraints, the iron sits outside the porphyrin ring. And this means we have iron 2. In the previous case, or if the iron is between the porphyrin ring, it's likely iron 3 or iron 4. But take a closer look. And one of the histidines is C-coordinated. And there's no reason for this because it's a really high resolution structure. So the chemistry is not correct. And I think it's a good point or a good time to bring up the point that good resolution does not mean good chemical accuracy and not even a good structure, because a chemical accuracy and structure, they are correlated with one another. Is this the only case? We look at the phosphatidylserase, completely different class of enzyme, two metal centers, an iron and a cobalt, 4 histidines, and of these 4 histidines, 2 are completely wrong. So 50% of the histidines are C-coordinated, which is chemical nonsense. So at this point, it's good to bring, we have errors of chemical accuracy. I take that chance to drink some water. AI is useful. So there are errors of chemical nature. We know them. But what's the impact of these errors on the PDB, on the AI models that we're training, structural biology, and drug discovery? Because at the end of the day, at our center, we are interested in drug discovery. What is the implication of a wrong structural model for drug discovery? And this brings us to the next point, which is the introduction of this new algorithm, the in-pocket analysis, which is our solution to this problem. So what is in-pocket? I'm going to use the benchmark case of axitinib bound to the BCR-ABL kinase to show how the algorithm works and what you can get from it. And you should think of these as a tool to test hypotheses. So it's a tool to test chemical hypotheses and to try to find the best model possible that suits the chemical or biological environment in a specific system. So what we do, we take a protein-ligand complex. We are usually interested in ligands, but it's not necessarily tied to a ligand. You can make the same question for a residue, a PTM, an ion, a water, doesn't matter, but we will focus on the ligands. So as a user, we say, I'm interested in these specific ligands. This is my complex. And what the tool does, it searches initially for all the residues in the vicinity of my ligands. And what we do is a series of experimentally biased quantum mechanical optimizations of the ligand residue pairs. But we don't do this only for the PDB deposited topology. We do this for all the chemical states, protonation states, tautomeric states, conformations that we identify and they are available to our ligands. Some of them might not even make so much chemical sense, like what you have here, but the tool is general enough so that if you don't have knowledge in chemistry, you can pose and ask the questions. So we do these QM optimizations, experimentally biased QM optimizations for all ligand residue pairs, all chemical states, and what we get is something like this. So we have a series of plots, one curve per chemical state, and these plots give us the ligand residue interaction energy for all the residues in the vicinity of the ligand, okay? So how can we read these? It's too many ups and downs, very, very simple. We put a line on 0, everything above 0 is bad, everything below 0 is good. Why above 0 is bad? This means we have clashes. Clashes are not physically, chemically, or biologically motivated. Therefore, they are bad. And we can look closer to this plot. We see red, blue, dashed purple, dashed blue. These all chemical states contain clashes. We exclude them as being unreasonable for the biological system we have at hand. And in this case, very simple, we conclude chemical state 5 is the most likely chemical state of the ligand inside the protein's pocket. If you compare with the PDB, that's what was declared originally. Okay, so there are differences. Ever since the first time I presented this work at the CCP4 developers meeting, there was a change, there was an update. And well, it's fortunate that we can still correct a few things, but yeah, so there are issues in the PDB and we can use this tool to identify those issues. Now you can see here why the chemical state 1 does not really work. And here we have a really nice figure because of Lucy, and she's there. We have the experimental density of the ligands, and you can see how well the ligand fits to that density. And the original topology deposited in the PDB, where you put a hydrogen atom here on this nitrogen of the pyrazole ring. And because you put the nitrogen here, you generate a clash with the main chain residue of the hinge of the kinase, and this basically generates the unphysical or unchemical motivated model. If you change the hydrogen from this nitrogen to the neighboring nitrogen, you remove the clash, you gain a hydrogen bond, and the model gets better. Now, is this everything we can do on this system? And the answer to this is most likely not. And to motivate what's going to come next, I would like to stress that this is a 2.2 angstrom resolution structure. And the question I have is, at 2.2 angstrom, can I really distinguish the orientation of this pyridyl ring here? Meaning, can I have it as it's oriented original on the PDB deposition, or can I flip it by 180 degrees and generate a better structural model? Now we have a tool to test this. We run in Pocket, we generate the new structural model, we run our calculations when we obtain the light green curve that you can see here, okay? And you can appreciate, on very brief looking, the light green is always below the dark green, okay? Meaning, chemical state 6 is supposedly better, has better interactions with the protein than chemical state 5. In other words, we prefer chemical state 6 over chemical state 5. The question is, how is the system going to behave? Are we going to have only one chemical state 6? Are we going to have a mixture of these two? And in order to answer this, we need the initial metrics, exactly. So this is what was missing. And the metric that we have is very simple. We sum all the ligand residue interaction energies. We add a few additional metrics, like the deformation energy, the relative tautomeric energy, protonation state, so on and so forth. And we get one single number that we call the in-pocket binding energy. Now, don't mistake this for a normal binding energy. This is just a metric, a fitness, how good the ligand or a chemical representation of the ligand fits inside the protein's pocket. And the numbers that we have for chemical state 5 and chemical state 6 are here. 63 kcal per mole for chemical state 5, 84 kcal per mole for chemical state 6. The difference is about 20 kcal per mole, absurdly large. We can clearly say chemical state 5 is not there. We have chemical state 6 instead. And if you're asking why, well, very simple. We have a tyrosine nearby. And the original model, you have a clash with this tyrosine, the hydroxyl group. If you flip the pyridyl, you remove the clash, you gain a hydrogen bond again. So these are the modifications. So what changed in overall? Well, we have this original topology of the ligands. We need to shift the hydrogen atom to the neighboring nitrogen, and we need to flip the pyridyl in order to get a better structural model. All of these modifications are in agreement with the experimentally deposited electronic density. So there's not much that could have been done except interpretation. So with this, we move to some downstream applications. And we're going to remain a bit on the axitinib BCR-ABL case, because fortunately for us, the deposition took place with two structures. We have 4WA9, which is axitinib bound to the unmutated kinase. And we have 4TWP, which is axitinib bound to the T315I mutant, which is clinically relevant. So what we can do is continue our simulations, assuming different topologies or different protonation states of the ligands, and see where they lead us. So we start with the PDB deposited topology. You can see here the differences between 4WA9 and 4TWP. And as a quantum chemist, I want to run calculations on the whole system. I do my calculations on the ligand kinase system, the whole kinase in this case. We can estimate the relative enthalpy of binding from our calculations, and we obtain a value of minus 23 kcal per mole. Now, I don't expect anyone to know what 23 kcal per mole mean, but you should think of these as some sort of an ITC experiment, okay? And you have two variants of the same protein, a single mutation, and the difference in the enthalpies of binding obtained from the calculation, it's basically 23 kcal per mole. If this is not enough, think about ATP hydrolysis, and the ATP to ADP costs 5 kcal per mole. This is 5 times larger, so absurdly large, okay? We can further process. We use the experimental IC50s to estimate the relative Gibbs free energy of binding. We back calculate an entropy, and we get minus 72 calories Kelvin mole. Again, absurdly large. we do the same experiments with the in-pocket topology. We lower the enthalpy of binding, or the relative, to -3 kcal per mole, more meaningful, and we obtain a relative entropy of binding of -5 calories Kelvin mole, again, significantly more meaningful than what we had before. So in other words, poor interpretation can be as bad or even worse than not having a structure, because it can guide you in the wrong direction. Now, at this point, I hope I was able to convey to you the importance of chemistry nowadays, because chemistry can enrich the structural databases. We have a protein-ligand complex, for instance, the one that you can see here. You have a nice ligand, contains an imidazole ring, and you don't see here, it's not represented, but in this pocket, we have 4 different carboxylate groups from glutamates, aspartates, and so on. And this is very interesting from the chemistry viewpoint. And we found this as an amazing case to test in pockets. And we run the calculations, and in pocket really tells us you should put a proton and have an imidazolium, not an imidazole, which contrasts with the PDB deposited topology. And this, I hope I can convince you easily, has potential value for drug discovery, has value for training AI models, and enriches structural databases. The problem is, and this is what I didn't tell you so far, that when I introduce this protonation, when I change the imidazole to an imidazolium, I also generate a clash with, I hope I can show, yeah, with the tryptophan that you have there, okay? And there's no way I can run my calculations, can add a proton and not generate a clash with the tryptophan, which it's a bit uncomfortable because it basically shows we need to put something, but we cannot put that something there. So given this situation, we went back to refinement, and Lucy did here an amazing job, and we concluded that this model does not exist. Very simply, what we have instead is not a ligand, a small molecule ligand. It's PEG, it's water, and it's zinc. And I hope I don't have to convince you that between a small molecule and PEG, there's significant differences. One has value, the other is completely useless for drug discovery. There's no value for training AIs, and it actually misleads the development. And this is, again, available in the database. So far, we focus on negative things that we can identify with our tool. It's already a good step forward, but there's also positive things we can extract. And in this case, we decided to run a series of calculations on one ligand, silmitasertib, that you can find here. This molecule is in clinical trials now. And 3 of its possible targets. We have 3 different kinases, CK2 alpha, and this ligand binds with subnanomolar affinity to CK2 alpha. We have then GSK3 beta, and the binding affinity is about 40 nanomolar, the KD. And then we have DIRC1A, and the binding affinity is about 1 nanomolar. The binding pockets are all very similar, so what drives selectivity? We ran calculations, we identified 4 different regions. We have, and these are all regions coupled according to how the amino acids engage with the ligand, so it's more or less ligand-centric. We identify first a no man's land, nothing really too special. There are some differences, of course, which can be exploited, but it's not really of interest for us. Then kinases, catalytic lysine. We see differences in how the ligand engages the 3 different catalytic lysines. In this case, it engages better the catalytic lysine of CK2 alpha. Then we have the hinge. The hinge, everyone knows, it's a very important binding element for kinase inhibition. And here we have something that is very interesting, because these 2 residues could allow us to shift selectivity between CK2 alpha or GSK3 beta, potentially. And last but not least, we have what I call here the selectivity histidine, because this is an element we found in previous studies to be critical for the subnanomolar affinity of silmitasertib to CK2 alpha, because the histidine basically performs a T-stack with this chlorophenyl ring. And if you remove this histidine, the selectivity goes down, or the affinity goes, it becomes less of a good binder. So we can benchmark our algorithm with previous knowledge that we have, but the question is also, how meaningful is this? And to prove the meaningfulness of all the calculations, we can run actual binding energy estimates using a whole chemical environment based on the in-pocket algorithm. And what we get is something that looks like this, the golden curve. And we can see that the binding energies reflect the experimental affinities to a very good degree. There's no correlation, but then again, with 3 points, it doesn't make much sense to put a correlation here. So I hope I was able to convince you with these slides or with this example that InPocket allows you to extract chemically consistent comparisons between different complexes, which can be potentially beneficial for understanding better how binding takes place. Now, at this point, I think it's fair to ask, are these isolated examples, or do you have more in your pockets? And this is an overview of some of the systems that we run calculations on. And here are many protein-ligand complexes. And in almost all of these cases, we had complaints of chemical nature. Something was wrong. Conformation, the tautomeric states, you name it, something was always wrong. We can also run calculations on cryo-EM structures, and it's not bound to crystallography or anything special. We need as input a structure, a structural model. The source doesn't really matter. With cryo-EM, in this case, we had 10 different complexes. We can run some statistics on it, which is nice. We also run calculations on protein nucleic acid systems. You can run a calculation using a single residue in the nucleic acid. You can include the whole nucleic acid. Of course, it's going to affect the computational costs, but it's still nonetheless manageable. And we run calculations on the proteasome, basically trying to understand the protein-protein interfaces. And of course, many issues we can find, for instance, carboxylate group and the histidine forming a hydrogen bond angle like this. So it's completely physically impossible. A few less words. InPocket was basically constructed or built using our in-house QM package, Ulysses. It's available as an independent Python code. You can just basically write us and we can take care of access for you for the time being. It's already fully implemented in CRIMS, so EMBL Grenoble. So if you use the high throughput crystallography facility, at the end, you can basically have the quantum mechanical validation, and the CCP4, PDB redo, and PDB integration is soon to follow. About capabilities, I showed you the ligand acid base and tautomerism. We can also run amino acid base. So you want to check what's the protonation state of a histidine, of a glutamate, glutamic acids, of a lysine, so on and so forth. You can test this using the algorithm. You can flip residues, so you can flip a histidine automatically. And we have now integrated the QM clash score, which is basically running using an electronic density and not just an overlap of the van der Waals radii of the different atoms. So this is a bit more refined. About efficiency, axitinib case, 10 different chemical states, protonation and tautomeric states, with 14 residues around, acid base and flips, 3 minutes in this laptop. So I think you might have resources for running the calculations. If I raise your interest, please make sure to check our preprints, and the submission is going to follow soon, hopefully. And with this, I would like to finish the presentation. I hope I convinced you that InPocket can be a useful algorithm for you to integrate in your workflows. It allows you to pose questions, to ask questions. How good is the chemistry behind my structural model? Additionally, it allows you to compare different models. This is an example I'm not showing you today, but if you look in the PDB, there's 2 different structures for curcumin inside the TYR2 kinase, and the binding modes are completely opposite. And we can use InPocket to basically determine which is the most reasonable binding pocket or the most meaningful representation of this protein-ligand complex. And because we do quantum, you don't care about which atoms you include. You can do almost anything, including transition metals. Another example is not shown here. The only thing is that the questions you ask matter. If you don't know how to ask a question, you might ask a garbage question and get garbage results. And this is very important, especially when you deal with transition metals, because if you don't have the coordination sphere complete, you will most likely screw up the whole system and the whole calculations, and you will not get meaningful results. Very simple example, a manganese, if you only have 3 neighbors, this is incomplete. You don't get meaningful results. And I hope that the presentation shows that structures without chemistry, they are still incomplete models, and we need to do more on this level. With this, I want to thank people, the Helmholtz people, especially Federico, who is supposed to be here and is helping further developing the algorithm. The guys from York, has been amazing collaborating with them, especially Lucy, who did all the crystallographic refinement and all that stuff. The guys in Grenoble, Jose Marquez, Kobe, Garib, a colleague of ours from Pfizer, and the guys from the PDBE. Of course, all of you for your kind attention, and a very special thank you to the organizers for having me here. And I will gladly answer any questions you guys have. Thank you.
Are there any questions? Yes.
Hi, Jose here from Diamond.
Yeah.
How much do you relax the structure of the protein itself to do this simulation?
So I basically apply a bias that fixes the heavy atoms to what you give as input. But it's not a fixed bias. It's not like zeroing the forces. It allows a bit of relaxation if the initial structure is not so good. Meaning, what happens is when you run an optimization, you keep the conformational state, but if you have stress in a bond's distance, in a bond angle, this is going to be relaxed. And the best example is nitro groups. All of the nitro groups are like a little Dumbo because the NO bond is overly stretched. And what we see in the calculations is that a shortening and to make the nitro bond symmetric, which is not always the case in the PDB cases, in the PDB examples we analyzed. So there is some sort of relaxing, mostly of the hard modes, stretching, the bending, and very little on the conformational state.
But you mean the actual residues?
It remains as if you exactly as if you have in the PDB with a small relaxation, given that you have a clash or something like this. If you have, say, a carbonyl group clashing an imidazole ring, it's going to go slightly away because the atoms are simply too close to each other and they start complaining, saying, "You're too close to me, go away."
Thank you.
Better.
Hi, Filipe. Here's Fia Cadia from
Hi.
Evotech. Thank you for your lovely talk.
Thank you.
You mentioned that you inspect the different protonations of the ligands, and that you just upload the PDB. My question is, do you take under account, for example, the protein buffer pH for the cryo-EM or the crystallization condition pH for the crystallography so that you're more close to what actually happens?
It's a parameter, the pH, but it's important to notice that the solution pH and the pocket pH might differ. So you always need to back check your results. So in that sense, you can change the pH as a variable. It's going to affect the protonation states that you find. But like I mentioned before, it's very important to consider this as a tool, not as something to tell you the truth. Okay, so you pose your questions, you know your pH better than no one else, or you should know it. And if the tool is basically telling you to predict something that is like a very strong base, and you know that you have acidic conditions, the results are not meaningful. So it's always a back and forth. It's questions, always about the question.
So you also mentioned that it's going to be delivered into CCP4.
Yeah.
Is it on CCP410 or is it in a future release?
I think it's that I cannot answer. I think John can give a better answer about the release.
Thank you.
Or availability. Sorry.
Yes, so we're working on it. We've removed all the showstoppers that we had. So, for instance, there was a problem with open bubble that has been replaced by Z-Kit. So it should happen this year.
Okay, I think that's the best answer. It should happen this year.
Okay, let's thank Filipe again.